Spun out of the Technical University of Denmark, Enduro Genetics addresses a fundamental problem in biomanufacturing: declining production at scale and poor yields.
I think you could say in terms of biomanufacturing, we take a little bit of the bio out of biomanufacturing and make it more manufacturing.
Yes.
So definitely, I think our technology is one of the solutions to sort of unlock the industry from being just specialty niche products to more everyday products and volume products. When you engineer an organism to produce the product that you want, it's a burden for the cell. And that's true for any type of cell, actually. And the way nature sort of is wired is that if it's a burden, it wants to escape. And that's what sort of evolution eventually also does. So the problem you have is that when you engineer an organism in the lab to make whatever wonderful bioproduct you have designed, then as you scale and as you move into to large-scale bioreactors, evolution kicks in, and as the biomass of the cells they grow, eventually over time they become less and less productive. New subpopulation is being formed in the bioreactor that figures out how do I escape, how do I produce less, and then they can grow faster than the producing cells, and then they take over the bioreactor.
Okay.
So that's a problem we address, and it's a problem that almost anybody in the industry has. Yeah. What we've developed is that we have a platform where we can make little genetic circuits that you can plug into an expression host, so an expression cell. And what these little circuits do is that they enable the cell to sense if it's productive, and then we link that sensing to the cell's growth. So the cell then becomes in a state where only if they produce, they can efficiently grow. And cells, they like to grow, so and multiply. That's what they want to do. So with our plugin, it sort of drives the cells toward high production and as they multiply. So there's sort of two effects when you plug in our technology. First and foremost, you have a better overall composition of high-producing cells. So instead of having a sort of a wide normal distribution of cells, you have a more tighter one towards the more high-producing cells right from the get-go. So that's the one thing. The other thing is that you have genetic stability over time. What can happen after 40 to 60 generations is that some of the cells, they figure out how to throw away the production gene completely and become completely non-producing. That doesn't happen with our technology. All cells will always produce at any point in time, no matter the scale. So you can run your production times for as long as you want, and that. And what that means is that you get more product out. Yeah.
So what kind of improvements have you seen in kind of titers with your approach then?
So if we take sort of a normal optimized process where the cell has been engineered and you have a process that works, and you run a normal fed batch process, we see around 30% improvement in titers. We also see other benefits in terms of it's more tighter, maybe with less biomass. So overall you have better productivity. If you have cells that are a little bit earlier in development time, it can be multiple times of improvements. Yeah.
So does this just work on bacteria? What about fungi, yeast? What testing have you done?
It works on all of the above, you could say. And this is really a universal truth in the sense that when cells are productive, no matter if it's a fungi or yeast or bacteria, they're in this state of high production, this burden state, and we can sense that, and all of them have essential genes where we couple these sensors to that regulates their growth. We went to the market about 2 years ago and also sort of followed the path of least resistance, and that gravitated towards mostly larger players with existing production processes and strains in place, where really they are, I wouldn't say in terms of ends meet in terms of optimization, but where they've already done a lot. And what you can then do with our technology is not to make the single cell better, just make sure that when you move it into a bioreactor, all the cells in the bioreactor are the best version of the cells. And that's not a technology that is otherwise available today with the other things.
So how do you actually implement this technology into a production strain? You talked about these tiny genetic circuits, and does it require any changes to the media or the production process, and any regulatory implications?
Yeah, so the way it's implemented is with standard genetic tools. So the one thing that you need to be able to engineer your organism. Then it doesn't matter really what type of organism it is, but you need to have the tools to engineer it. And these are standard tools, and we have tools, and the clients have tools, and these things, and also you could say off-patent tools, so you don't have licensing complications in terms of working with our technology. And then what we do is that we've optimized the plugin to your existing process. And that also means that you don't need to change your existing process. It's actually tailored to your existing process. You can start playing around with your existing process because you might have suddenly have a sales competition that is actually more hungry to produce more. So you can increase the feed rates. There's other things you can do. You can extend the fermentation runs if you want to do that. You can do fill and draw in terms of so you save for some time, or you can look towards continuous. Also, you can do that. So you have more freedom to work with your process, but you don't need to. You can plug it in today and already get benefits.
And what about? And are there any regulatory implications?
Yeah, there is. And they can be, depending on what industry you're in, it differs a little bit, but if you sort of take a standard bioindustrial process, it could be a food component or another type of ingredient. What the clients have today is they've already have the regulatory approvals for their strains. And when you implement our technology, you don't introduce any foreign DNA or genes. It's all built with the cell's own genes and own DNA. So the regulatory implications are very light. In pharma, if you're looking at pharma implication, it's a little bit different.
Are clients kind of worried about letting their proprietary strains kind of leave the premises? Are they kind of, I'm sure they're not kind of saying, oh, here, just take our strain and play with it.
Some are and some are not.
Yeah.
And we address that in different ways. We don't need to have the strain in our facilities to do it. We can do it in a way where the plugins, we actually deliver them as a data file, as sequences that they can then introduce. But we can also take the strain to our laboratories. We have secure laboratories in Copenhagen, and then we work with the client strain.
But if they have the expertise, they can actually do this transformation
They can
themselves.
do it themselves, and sometimes it can be better because we don't. Then we don't have to do tech transfer of protocols and different things, which it takes time, it's costly, and it's a risk.
Okay, so what progress have you made to date, and when do you anticipate potential commercial implementations of this technology anyway?
Yeah, so we have a little less than a handful of commercial clients today that we're working with, and we've been able to build that base over the last 18 months, 2 years, I would say, across bioindustrial and across pharma, and we will deliver the first 2 projects this year. We take a technology access fee to get access to working with us and where we design these plugins for the clients. Then if we implement it, we do that as a service. So that's a cost plus model where we sit down with the client and agree on the split of tasks, and then we do that. Then there's a success milestone payment when we've done it, and then there's a licensing model where they pay us annually based on the use of the technology. Some clients also want to buy it out completely, and we can also do that.